Hello, everyone, and welcome to this webinar on discovering scientific evidence using Europe PMC SciLite annotations. My name is Diane Araújo. I'm part of the team work on the Europe PMC project at the European Bioinformatics Institute, and I'm here today with my colleague, Aravind, who will help me to answer any of your questions at the end of this session. So we will start with a brief introduction to Europe PMC and the annotation platform. We will then go on and focus specifically on how to use Europe PMC SciLite annotations to optimize your reading process and find evidence in research literature. And finally, we will show you where to find the related information and how to get support if you need it. And at the end, we will try to answer any of your questions. So Europe PMC is a free global database of biomedical and life science publications. It's developed by EMBL-EBI and is a partner of PubMed Central. Europe PMC is a repository of choice for 31 international science funders. And it offers comprehensive content such as abstracts, full-text articles, preprints, patents, agricultural records, and NHS guidelines. Europe PMC is more than a place to get a free paper. Open access is just the first step. There is so much more. Publications are enriched with citation network, data crosslinks, text-mining annotations, ORCID IDs, and biomedical grants. Let's now have a look at Europe PMC annotation platform. At Europe PMC, we consider literature as an important part of big data in biology. There are currently over 37 million abstracts in Europe PMC. Journal articles are the main means of dissemination of biological findings. Scientific articles contain important information and data extremely valuable for knowledge discovery. Scientists access this information by reading articles. With the tremendous growth in the rate of publication, it is hard for researchers, curators, and scientists to extract all the information contained in these articles. To effectively extract information from such a large number of publications, automation is an essential component. So here, text mining, it's often used to extract knowledge. However, its application can be limited due to the technical know-how to set up a text mining pipeline. At Europe PMC, we want to help bioinformaticians, data scientists, and researchers to overcome this hurdle. Therefore, we have set up an infrastructure to host text mining outputs or annotations. This way, annotations from different sources can be aggregated and shared with the larger scientific community. Currently, the infrastructural components includes an annotation platform to integrate text-mined and curated annotations, and an API to access all the data housed in the platform. So also we have an application called SciLite that's used to highlight the annotations on article pages in Europe PMC. The annotations are on all abstracts, open access, full text articles. And latest component of this infrastructure is the annotation submission system, which is an easy way to submit text-mined and curated annotations to Europe PMC. So what are annotations? What we call as annotations are basically concepts identified by either a text mining algorithm or by a human curator. These concepts could be chemical or protein name mentions, and the target concepts or annotations are linked to a relevant database. Europe PMC Annotation Platform currently provides access to over 1 billion annotations to nearly 20 different types of annotations, including genes, proteins, phenotype, organisms, chemicals, gene disease relationship, and metagenomics annotations. So these annotations, they are provided by a variety of text mining groups. They use text mine algorithms to identify different types of biological entities. There are two ways to access the annotations data in Europe PMC. The first one is the programmatically access via Annotations API. You can find the details on the API under the developers section of the Europe PMC website, or check our webinar Extracting Research Evidence from Publications, which link is provided on this slide. The second way to access annotations is via the Europe PMC website with the SciLite Annotations tool. SciLite is a tool that highlights biological terms or relations identified by text mining on abstracts and full text articles. It helps you to read the paper faster, focus on important concepts, and to find data and evidence within the publication. So let's see how it works. When reading a paper in Europe PMC, on the right-hand side, you will notice a menu titled Annotations. You will then be able to select the concept of your interest. So now let's play a short video here. You can select the concept individually or also select all the annotations from a specific type to be highlighted in the text. And also you can use the navigation buttons to jump to select the terms in the text. So among annotations that you can select are biological entities such as gene and protein names, organisms that corresponds to species names, chemicals, disease, and gene ontology terms. For those unfamiliar with gene ontology, it is a controlled vocabulary that defines concepts describing gene function. Membrane, transcription, catalysis, those are some examples of gene ontology or GO terms. You can also find annotations for protein phosphorylation, gene function, phenotype, as well as relationship information such as gene disease associations and protein-protein interactions. As I have already mentioned, the annotated concepts, they are identified by text mining algorithms. Some of those are developed by the Europe PMC team, and others are submitted by other collaborators. For example, gene disease relationships are contributed by two groups, Open Targets and Diganet, and unlike other annotations, protein-protein interactions are manually created and are provided by Intact. So how you can see SciLite to help you with your research. If you need to quickly scan the article for keywords to understand if it is relevant to your topic, or if you would like to skim read the paper, focusing on the important bits without having to read the entire article, you can simply switch on the highlights for annotated concepts. In the example here, you can see the excerpt from the paper manually discusses CARF protein and WNT signaling pathway in mice reproductive system. SciLite can help you to see what are the main players described in the text, which model organism the work has done in, and what kind of drugs are used for treatments, and so on. You will notice that some of the terms could be listed several times. For example, Mycobacterium tuberculosis and M. tuberculosis might be referring to the same organism. However, the TextMine algorithm will list them as separate entities. Nonetheless, the list can give you an idea of the most common concepts appearing in the text. Notice the number in brackets next to the annotation name. It indicates how many different terms belonging to that group were found in the text. Europe PMC has teamed up with Magnify, an EBI genomics resource in the Emerald project to identify metagenomics concepts in Europe PMC literature. Those concepts cover entities related to biome, sample, and origin, as well as metagenomics experimental methods, and are all available in Europe PMC Annotations platform. 16 metagenomics concepts can be highlighted in the text and open access articles using the SciLite Annotations tool. Highlighted concepts include sample material, body site, host, state, date, place, treatment, and library strategy. Sometimes, despite stringent quality checks, you might notice that an annotation is too generic or incorrect. It is of critical importance that readers find the annotations useful, so you can provide feedback on each annotation. Again, I will play another demonstration video here. Hovering over the highlighted terms, it will bring a pop-up window where you can endorse annotations or report if the term is too generic or incorrect. This information is fed back to Europe PMC team, and we will act on that. So you can also use the share option to get a link back to this specific annotation. While reading a publication, and depending on your background, you might find some unknown concepts in a paper, or perhaps you might need some additional information about a familiar compound. SciLite annotations are linked to structured records in public life science and chemistry databases. So again, I will play a short video here. If you hover over the highlighted terms, it will bring a pop-up. I will pause it here for a moment. So it will bring a pop-up menu with a link to associated data record. So for example, when clicking on this compound here, this chemical molecule, it will be you will be able to visualize the chemical structure, and you will also get a link to the Dictionary of Chemical Entities of Biological Interest. So playing here again, if you follow that link, you will see a summary dedicated to this particular compound in the database. So SciLite annotations include functional relations such as gene-disease associations and protein-protein interactions, which can help you to find evidence and formulate or refine your hypothesis. So for those annotations types, an entire sentence that contains the evidence for the relation will be highlighted on the text. So here there is one example where we find evidence that E2F1 transcription factor interacts with P18NK4C promoter according to a chromatin immunoprecipitation experiment. In the second one, we find information on how the interaction of E2F1 and the P8 promoter relates to human glioblastoma tumors. So let's again play another video. Here, if you click on the annotation again, you see a pop-up menu. They will link to the resource with additional description. So for gene disease associations, you will find two links displayed on the pop-up menu. So one for the gene and another one for the condition associated with it. So let's play it again. The first one will link to UniProt, and the second one will link to the EBI ontology lookup resource. So if we just keep an eye on it, you will see how it links to each of the data resource. Here to UniProt, and now here to EBI ontology lookup resource. So finally, there's one more function that SciLite can provide to support you in your research. It enables scientists to locate the primary data from the text for reference result or test your hypothesis. By using public omics data, you can focus on accession numbers in the text that link to the primary data. So accession numbers are unique identifiers given to a data record to track it in a single data repository. Such identifiers are crucial, especially because proteins and genes names, they are often ambiguous. So accession numbers are yet another type of SciLite annotations. You can also highlight the accession numbers on the text. Let's play here again. So here in this example, you can find reference for PDB records for protein structures in the text. You can see the structure viewer. You can interact with this protein structure, as you can see here. And you can also find the link to the data resource, to PDB-E, to check this protein structure in more detail if you wish to. SciLite tracks accession numbers for several different resources, including Array Express, UniProt, Pride, PDB-E, it's the Protein Data Bank Europe, European Nucleotide Archive, and many others. So you can see the full list on this slide. When using a SciLite tool, you will notice that the full text of some articles will not have the option to select annotations on the menu on the right-hand side. So although we see the list of annotations, you cannot highlight them on the text. This is because the use of material is subject to the copyright and related license terms of the respective authors or publishers. So SciLite annotations can only appear on the full text article with a CC BY NC or CCO license. So we are now approaching the end of this session, and I would like to share some contacts on Europe PMC before we get your questions. If you have questions about Europe PMC, please contact us on helpdesk@europemc.org. If you text mine or create biological concepts and you wish to provide them to Europe PMC Annotations platform, please contact annotations@europemc.org to give us more details and discuss that. You can also watch our webinar, How to Share Text Mining Results in Biology, that gives you also a tutorial on how to submit text mining or created annotations to Europe PMC. And you can also follow us on Twitter to get updates on new releases. So now I will hand over to Ajay.
Right. Thank you, Diani. Thank you for today's webinar. And thank you to all our participants for joining in. So with that, I'll open the floor for questions. So if you have any questions, please do type them in the Q&A box. So Diani, so we have a question in the chat box. So can we add annotation to enhance the performance?
Yeah, I would pass this one to Arvind, since he's the one managing our annotation platform.
Thank you, Diani. I'm not sure what you mean by performance, but we don't do, within the platform, we don't do any sort of benchmarking per se of the text mining outputs. We leave that to the providers to benchmark their annotations themselves. However, if you're interested in general in the academic part of improving performance, you could always download, access the annotations from our API, and then and maybe contact us, and then we can proceed further. I hope this has answered your question.
All right, so if we have no further question, I would just like to tell you that the recording from this webinar will be made available very soon. So thank you very much, Diani and Arvind, for today's webinar, and thank you to all our participants for joining in today. Thank you very much.